a friendly college community where the wind is strong, the moon is beautiful, and mysterious lights illuminate the campus while we all pretend to study. Welcome to Yukon. This is WHUS Stores.
Hi, my name is Lady Mystique, and I am a radio show host here at 91.7 WHUS Stores. I have always loved all kinds of music, and I believe it has the power to bring people together and to uplift them no matter what they are going through. And I am adamant about supporting local artists by having these artisans on my show for on-air interviews. Additionally, I believe we can all use some help with our relationships, so I like to share the knowledge and strength I've gained from my past experiences with my listeners, and I encourage them to do the same with me. I also like to throw in a little bit about astrology because it never hurts to know who you're dealing with, right? You can catch my live show right here every Saturday afternoon from 1 to 4 p.m. Eastern Standard Time on WHUS Stores.
Hello and welcome to DNA Today. I'm your host, Keir Deneen. DNA Today informs you on what's happening in the genetic world. The show is packed with news stories and lessons, all to keep you updated and educated on what's happening in genetics, especially what's happening at UConn. Today we have some news stories about malaria, genetic roles in stress and heart conditions, a new method of identifying and analyzing DNA, and we're also going to learn a little bit about Huntington's disease. I like covering some sort of a lesson. I don't want to overwhelm you too much, so a nice, easy lesson. So let's delve into the genetic roundup of the week. Our first story, as I said, is about malaria, and a collaborative international study on human genetics of malaria has revealed new insight into the susceptibility to severe malaria. So there's 5 genes that they've identified as being linked to having a complex role in either protecting or making people more susceptible to severe malaria. So really, one way or another, either you're protected from it or it's actually making you more susceptible to getting malaria. Now, this information was published in the journal Nature Genetics, which I refer to a lot because it's a genetics show and it's an academic journal. Definitely cool to check out if you can actually read it and understand what's going on. A lot of times it's easier to read more of a watered-down news story. But before we get into all the details of the genetic portion, let's back up in a moment to look at what malaria actually is. Malaria is a disease that develops when a mosquito infected by the parasite Plasmodium bites someone. And through this bite, the parasite invades and lives in the person, the host's, red blood cells. It can take a couple days for the parasite to manifest itself, and it is a very serious disease, as 20% of the patients with malaria die despite being treated at a hospital. It has the potential to affect the brain, which is why it's such a serious disease, because anything that's brain-related is affecting your entire body. And if the brain is affected, it can cause a coma or cerebral malaria, which I'll refer to later, so you can remember that. Other vital organs, such as the kidneys and lungs, can also be affected, and even the blood. So it really is affecting a lot of areas of the body and different body systems. Dr. Sarah Dunson of the National Institute of Global Health at the University of Melbourne in Australia was heavily involved with this project, and she, along with her colleagues, analyzed data of nearly 12,000 cases of severe malaria. So they definitely had a lot of data to work with and a lot of case studies. These samples were collected all over the world from 12 sites, including Africa, Asia, islands around the Pacific Ocean. And these areas were access to treatment facilities. And that can be a difficult thing because you have to have the right connections, and especially in places that are more developing. And that's another reason why this study is so important and why I'm talking about it today. So Dr. Sarah Dunson shared, quote, "It involved a large number of severe malaria patients from multiple countries, which allows us to identify genes that truly have an effect on whether or not you develop severe malaria." So by gathering this large amount of data, it was easier to investigate genes that influence susceptibility to malaria on an unprecedented scale. So having so many cases, you can really see if a lot of the cases and a lot of the genes that are a lot of the genetics, chromosomes analysis, all of that, if a lot of genes are matching up, or specific base pairs, I'll get into that a little bit later, are matching up, then it's more of an indication that it actually does have an effect on malaria if we're comparing it to a normal genome. Something that's similar in a lot of them people with malaria that is not similar in people that don't have malaria is an indication that those genes have an influence on malaria. And as I previously stated, the team found 5 genes linked to malaria. And this is out of the 27 genes they analyzed. So 5 out of 27 ended up, they believe, are linked to malaria, and the data supports this. And their findings were not completely clear, though. Dr. Dunstan commented on the complexity of the genes. Quote, "Our findings revealed that deficiency in one of the genes, G6PD, which causes a genetic blood disorder, can both reduce risk of cerebral malaria and increase of severe malaria anemia," so anemia blood, "both of which are fatal complications of malaria." So this is one of the genes that has a dual effect, where it reduces the risk of cerebral malaria, but at the same time increases the risk of a different type of malaria called malaria anemia, which is when the malaria affects the blood. And cerebral malaria is the brain malaria I mentioned earlier that I was like, remember that. So this research is really exciting as it leads to new findings, bring us closer to understanding how immunity against malaria works. And if we're finding genes that are, or types, alleles of genes, types of genes in people that have malaria versus people that are maybe immune to it, seeing those differences can really pinpoint to which genes are causing someone to be susceptible of malaria, whereas other people are immune to it. So obviously always learning more of this information helps coming into more treatment research and always eventually, hopefully, that cure. Our next news story, Duke Medicine has published new findings suggesting that some people who are prone to hostility, anxiety, and depression may also be hardwired to gain weight when exposed to chronic stress, which leads to diabetes and heart disease. So knowing that you carry this gene can help reduce heart disease with straightforward preventative measures such as healthier eating, working out, and managing stress levels. So by saying, you do have this, maybe it will give that person, that patient, more of a motivation to really take care of themselves so that they can prevent things like chronic stress, diabetes, and heart disease. And Dr. Elizabeth Hosser, the director of computational biology at the Duke Molecular Physiology Institute, shared this, quote, "Genetic susceptibility, psychosocial stress, and metabolic factors are act in combination to increase the risk of cardiovascular disease." And her colleagues analyzed genome-wide association data from nearly 6,000 people who were enrolled in the multi-ethnic study of atherosclerosis. And this study began back in 2000, so 14 years ago, and with the goal of understanding how heart disease begins. And this is explored through the collecting of participants' genetic makeup and physical measurements such as body mass index, glucose levels, cholesterol readings, among a lot of other things. And in this, at first, researchers identified as a strong correlation between participants reporting high levels of chronic stress and increased central obesity. So the more stressed someone is, it seems to be that there's an increase in obesity. And the researchers then tested genetic variations in the genome to see if there were genetic variations when combined with stress resulting in higher central obesity. And the variations I'm referring to here are called single-nucleotide polymorphisms, which I believe I've mentioned a couple times on the show so far. And they're known as SNPs for short, so maybe you know it by that. And this is where a single base pair, A, C, T, or G, adenine, cytosine, thymine, guanine, are different in particular locations of the DNA when compared to like the general public or like, a healthy genome or something like that. And so it turns out that people had this correlation between chronic stress and central obesity, had these SNPs, single nucleotide polymorphism. So one single base is changed. And all these people that had this correlation between stress and being more obese, they had this one gene in common. And the gene in common, they had the SNP was in common in this gene. That's a better way to phrase it. The SNP in this gene was in common. And the gene is EBF1 gene. And the researchers measured central obesity with hip circumference, and they found a very strong correlation with hips growing wider as stress levels increased. So Dr. Singh, a researcher in computational biology also at Duke, and the study's lead author, shed some light on this portion of the study. Quote, with further analysis, we find a significant pathway from high chronic stress level to wide hip circumference, to high blood glucose and diabetes, to increased cardiovascular disease. But we found this only in people who were carriers of this EBF1 single nucleotide polymorphism. And this was limited to participants who were white. So, end quote. And so he really, this research is very strong and very clear compared to the last news story we talked about that was a little bit, sometimes the person either would be susceptible to malaria because of a genetic variation, or they're immune to it because of genetic variation, where this is very straightforward. If you have this base pair that is different in this one gene, then it's very likely that, this correlation applies to you, that if the more stressed out you are, that you have this chronic life stress, that you're going to be more obese, and a slew of other things that I've mentioned. A senior author of the study and director of Duke's Behavioral Medicine Research Center, Dr. Redford Williams, shared, quote, these findings suggest that a stress reduction intervention along with diet and exercise could reduce the risk of cardiovascular disease and may be most effective in individuals with this specific genotype. So by actually lowering stress in non-traditional medicine senses of, maybe taking more breaks, exercising, things like that, can actually really improve someone's life and can improve their obesity and just their health overall. So it's coming down to, we can see that someone is genetically different and predisposed to have these things. And because of that, we can say, look, you have, hopefully, we can say down the road that you have this gene, you have this variation, this SNP. If you follow this, then this kind of exercise plan or diet plan, whatever, kind of increase in health, then you can avoid all of these side effects from it. And Dr. Elizabeth Hauser, who I mentioned before, again, the director of computational biology at Duke Molecular Physiology Institute, concluded that, quote, we need to figure out how these genetic factors influence the increased accumulation of fat in the central body and increase the body glucose levels in persons exposed to high life stress, and why they're also different with ethnicity. This knowledge could help identify targets for behavioral and drug interventions that could reduce disease risk. So now that we found this correlation between this gene and being more obese when exposed to stress, now we have to figure out why that's the case, why this gene, like what it's actually doing. And once we do that, obviously it will help out on figuring more medicine out. And the more we know, the further we go, right? All right, this next news story is really cool and has the potential to change the way we use DNA to profile people. Researchers at Johns Hopkins have been developing an improved means of detecting mismatched DNA. And this new method has the ability to analyze tiny amounts of DNA. This could help forensic scientists match DNA found at a crime scene to the criminal. Another use it has is in organic transplants. Donors would not have to go through invasive testing to ensure their organs are a match, and lots of other different methods. And researchers have applied for patent for their work, which is something to note. And currently, the number of specific repeating sequences not in the genes is the way DNA is used for identification. So by looking at, you have a certain number of base pairs that repeat, by looking for that is different in people, and that's how they can identify if DNA matches someone. And so Dr. James Eichmann, a professor of pathology at the Johns Hopkins University School of Medicine, explains why this method is not sufficient enough. Quote, repeat testing will only detect DNA that makes up at least 1% of a DNA sample, so it's not great for situations which results depend on small amounts of material within a larger sample. So if you're at a crime scene and you just get a speck and it's not really a lot to work with, it's very hard to use this repeating sequences method to identify the DNA and match it with someone. Whereas this new method, it's a little bit more promising and it doesn't need that at least 1%. It actually only needs 0.01%, so big change. And in the past, identifying DNA through point mutations within genes was too expensive. So by looking at, like I said before, the SNPs, single nucleotide polymorphisms, can differ between people, and that's really what makes people's DNA different, because most of it is the same, very, very high percentage of it. And so in the past, this has been too expensive to work with using these point mutations. But with new technology, sequencing DNA isn't nearly as much as it used to cost. You can get it for a couple thousand now, whereas it used to be like 100,000. Not even that long ago, really. And Maria Debeljak, a technician in Eichmann laboratory, used this method on this point mutation method, and she said, quote, we could detect cells when they made up just 0.01% of the mixture, which is a big improvement over the current method, which can only detect DNA that makes up 1 to 5% of the sample, end quote. So therefore, there's not much DNA needed in this mixture to be identified, and that's really the plus about this, is that it's more efficient in terms of how much DNA is needed to actually be identifying this and matching it to other people. And the more efficient we are, the more we can actually be identifying DNA. And I'm sure in certain situations you couldn't identify DNA because there wasn't enough of it. So with this new method, it's definitely promising to be able to use it. And it's not just matching people to crime scenes. This new method can be used for a lot of other things. It can be used as a paternity test, organ donor test, as I said before. Even the news story I got this information from the genetictimes.com mentioned that it can monitor the health of bone marrow transplant patients by testing transplant patients with blood for low levels of leukemia blood cells, and therefore they theoretically could be used as an early warning sign of leukemia. And so there's so many different things. It's not just matching people with DNA to see if it is them. It's a lot of things that if something is compatible with each other, like the organ donor test, predicting things like with leukemia and blood cells, paternity test, that's a little bit more of, does it match kind of thing. And so there's a lot of different ways that this can be used, and that's just to name a few. I'm sure there's plenty others that are very, very complex and all of that. So that's it for the news stories of the week, a little bit of a roundup there. And something to note before I move on, before I forget, Wednesday is the opening of the Jackson Laboratories in Farmington here in Connecticut, not too far from UConn. And so I will be attending there on Wednesday. I'm very excited to hear all of the really thought leaders in the industry of genetics, and just all day hearing them. So it's be nice to get off campus and hear from real professionals, and I'm sure I'll have a lot to talk about next week reporting on it. So if you can go, I'm sure there's still spots open. They had spots open last week. I'm not sure about now, but definitely check it out, and hopefully they upload things online so you can maybe see kind of a TED Talk kind of thing. But yeah, definitely keep your eye out. I will definitely be tweeting throughout the day there, and hopefully some pictures here and there too. All right, so the genetic disorder I'm going to talk about this week is Huntington's disease. And this is a disease of the brain, and it affects the nerve cells in the brain, which then affects behavior, movement, and cognition. It really affects everything, as a lot of brain diseases do. And this disease is caused by a mutation in the 4th chromosome. In healthy bodies, this gene produces the protein Huntington, and which makes them function poorly and die. So, I'm sorry. So, in a body affected by the disease, this gene is larger and produces a larger form of the protein. So, in a normal body, it produces Huntington, but in a person affected by Huntington's disease, the gene is larger and therefore produces more of the protein. And cells in the brain are very sensitive to the effects of abnormal, larger Huntington protein, which makes them function poorly and then die. And in most cases, people begin to show signs in their 40s and 50s. People that show signs earlier in life, 20s, are identified with juvenile Huntington's disease. And symptoms include movement disorders of both involuntary movements and can also impair voluntary movements as well. And the cognitive aspect of the disease, it can impair thinking, judgment, awareness, and perception. Depression is also a common symptom when affected by Huntington's because the brain is changing and injured, so that is affecting, I would guess, different levels of different chemicals and probably dopamine, serotonin, which leads to happiness and all of that, feeling good. There's a 50-50 chance that a child of a parent carrying the Huntington's disease gene will develop the disease as well. And there's no cure for Huntington's. There is medication which can help with symptoms and kind of ease that and treat, but there is no slowing it down. It kind of just, it makes you feel a little better, but it's still going to take its course. And kind of talking about the history of learning about Huntington's and how we kind of came about finding it through research. It was kind of a surprise breakthrough. In 1983, a genetic marker was discovered which localized the Huntington's disease gene to an area close to the tip of the short arm of chromosome 4. Before I mentioned that the disease is linked to gene on chromosome 4. And from pinpointing the location of the gene, genetic tests became available to find out if someone had the disease using a method called DNA linkage analysis. That's DNA linkage analysis. And this test began in 1986. Now we're 3 years later. The genetic marker was found in 83. And 3 years later, in 86, the testing began in Johns Hopkins. Second time mentioning them today. But not as efficient and accurate as it is now. Before, multiple family members had to provide DNA, and it was only 95% accurate at best. So you couldn't even get a clear test result and answer from it. But as time went on, more genetic markers for the Huntington's disease were found, and testing became commercially available. So in 1993, so now we're 10 years after the genetic marker was found. That was in 83, so now 93. The gene was announced as being isolated, so we clearly identified it. And the mutation of the gene that causes Huntington's disease is actually a pattern of 3 DNA bases. So remember, the bases that I mentioned are adenine, guanine, cytosine, and thymine. And those are just the bases that exist for DNA. But the 3 bases that affect Huntington's disease is CAG, cytosine, adenine, guanine. And that repeats more often than the normal version of the gene. So this is something the CAG would repeat over and over. And this happens more in a chromosome from someone that is affected by Huntington's disease. And because it's more prevalent, because there's more of them, more of the protein is produced, and that's what causes all of these different symptoms that I've referred to. And there's no correlation between the number of repeats and the age in which the person is affected. So if someone has, we're just going to use total hypothetical numbers, if someone has 50 repeats compared to 100 repeats, the person with 100 repeats is not necessarily going to have juvenile Huntington's disease. It doesn't quite work like that. There's no correlation behind that. And with all this testing becomes the question, should I get tested? If you have family members you don't know, is that something that I want to do? Do I want to know when should I do that? When's the best time in my life? When I know, am I going to change decisions later on? Will I make more rash decisions or do things earlier in life than I probably would have? And it's the decision to be tested is a very personal one that someone has to make themselves. It really has to be their decision. But the daunting task and choice of, should I do it or shouldn't I, can be made easier with the help of a genetic counselor. That's what they're there for. And a genetic counselor's job is to educate the patient so that they can make a more informed decision. it's also the genetic counselor's job to remain unbiased and present the information without their opinion, that here's what you can do, here are all your options, and make sure that the patient knows about how each option works. And from that, then the patient can make a decision with their genetic counselor. And the role of genetic counseling is very important because doctors don't have as much time to really sit down with patients and go through their family history, go through the options, what they feel like they should go with, any questions they have. They actually have time to sit down and answer all those, and it's typically, hopefully, not a rushed kind of meeting where they can feel relaxed and really go through everything and make sure all their questions are answered. So that's why, genetic counseling the field is really expanding and the job opportunities are fantastic because there is more genetic testing, and because genetics is such a rapidly growing field, that genetic counseling is definitely, if you're interested in genetics, definitely check that out as a career option. I'm kind of looking at it myself. But yeah, it's a very hard decision to make whether you should get tested or not, especially because there's nothing if you know about it, you can kind of plan your life accordingly, but there's no way that then you could have a procedure done or, a surgery done to prevent it from happening. Unlike things that we've talked about before of genetic testing of breast cancer. If you have the BRCA1 gene, if you test positive for having the mutation in the BRCA1 gene, then you would have that decision if you should have a mastectomy or not. But with Huntington's, you kind of just know, and it can help plan your life accordingly and everything, and just children, marriage, all of that, life events. But it's definitely a hard decision to make, and that's why genetic counselors are so important. So that's just a little bit about Huntington's disease. And I think we have a little bit of time. I could throw this in here. So I thought I'd teach you a little bit about bioinformatics and kind of what that is. And as scientists are discovering new advances in genetics, they need a system to store this data and be able to have easy access to it. As technology advances, this becomes easier as computers can do most of the organizing work. So knowing this, bioinformatics is this system of using computer technology to manage all this biological information, because as I've been mentioning before with news stories, there are so many, thousands upon thousands of just in one one study looking at different people's genetics and all that, and you can't just have that in a file cabinet. You need it in some sort of a database, and bioinformatics is making that work and making it happen and just managing all of this biological information. And this field is exploding right now due to the exponential expanding of the genetic field, as I keep talking about it and I'm so passionate about, resulting mainly from the Human Genome Project. And the Human Genome Project set out to sequence human DNA, all of it. So their goal was to make a map of the genes in our body, and they're between 20,000 and 25,000. So this obviously was a huge project. The first draft of the gene sequence was completed in June 2000, and the project was declared complete in April 2003. And you may be asking yourself, humans don't have all the same DNA, so how are scientists mapping the human genome? Excellent question. Mapping the human genome involves sequencing multiple variations of each gene. So since, as you pointed out, we don't all have the same genes, so you have to include the different alleles for genes. Again, most of it is the same, but you do have that small percentage that does differ. So why am I talking about all this? Where does bioinformatics fit into the Human Genome Project? Well, as I said, scientists in the bioinformatics field use computer technology to store and analyze all this biological information. You can't really put it, on a piece of paper and figure it out yourself. We really do rely on computers now to figure all of this massive information out, all of this big data. And when the Human Genome Project was beginning, a major concern was how all this information would be stored and accessed. And database is an obvious answer, but designing one that fits all the requirements is easier said than done. And one vital requirement was for researchers to access data and submit their own. It has to be a two-way thing. And many people use computational biology and bioinformatics interchangeably, but there is a difference. So I just wanted to kind of go over this. Computational biology is using computer techniques for the study of biology. The goal is to acquire knowledge about living systems. It focuses more on the science aspect. Now, on the other hand, bioinformatics is the creation of tools such as databases and algorithms to solve problems. The goal is to build useful tools for biological data, and it focuses more on the engineering aspect and the computer science part of how to make that happen. So that's really focused on the computer part, whereas computational biology is using the technology that bioinformatics produces and develops to learn about living systems and to add their information too. So bioinformatics kind of comes first and then computational biology, but a lot of times computational biology, what they're doing can influence bioinformatics. So it's both the relationship is very important to each field, and it's a very tight relationship. And the tools created in the field of bioinformatics, again, can be used for computational biology. And they are both managing big data and providing scientists access to this fountain of knowledge. Well, glad I got to fit that in. Just barely made it. All right, so that wraps up today's episode of DNA Today. Tune in next week, Mondays at 1 p.m. and every Monday at 1 p.m. to learn more about genetics on DNA Today. The website is dnapodcast.com. And if you want, you can follow on Twitter at DNA Podcast. And you can follow me on Twitter at Kiradineen, Kiradineen. Shoot me an email if you have any at info@dnapodcast.com. Thanks for listening, and join me next episode to learn and discover new advances in the world of genetics.
This is Pack 15. You are listening to
WHUS!
Muy buenos días, compadres. Si quieren escuchar y disfrutar de música latina, música brasileña, ritmos gitanos y africanos, asegúrate de sintonizar el próximo episodio de Yerba Mate, los sábados de 8 a 10 de la noche. Hey there, Radioland. For the best Latin jams and foreign folk, be sure to tune in to Yerba Mate, Saturday nights from 8 to 10 on 91.7 WHUS Stores.
Hey, Mr. Lebowski, what you listening to?
I am not Mr. Lebowski.
I'm pretty sure you're Mr. Lebowski.
I'm the dude. So that's what you call me, you know?
All right, dude, if that's what you're looking for. I didn't mean to step on your toes.
You're Mr. Lebowski.
I don't know about that. I think you might be wrong there.
Hey, at least I'm housebroken.
That's a low blow, man. You said you'd never bring that up again. So, hey, not to inorganically transition or anything, but what do you think about regular corporate radio?
Treats objects like women.